welcome. This is a PowerPoint presentation going over the causal criteria, sometimes referred to as the Bradford-Hill causal criteria. And I'm going to start with just giving the list. This is the list that's in the book, Rothman, Strength of Association, Consistency, Specificity, Temporality, Biologic Gradient, Plausibility, Coherence, Experimental Evidence, and Analogy. So the first thing to know is this is a list, but it's not meant to be a checklist. These are arguments that we use in epidemiology to formulate and understand results to get a sense of the concept of causality. So what is a cause and how do we establish causality? Well, the first thing to know is that we cannot really know the cause for sure. So in epidemiology, what we see are relationships. We see associations. If we wanted to truly establish causality, we would need an event or exposure that we could observe directly and that we could see the chain of events that results in the outcome. So how do we actually talk about causality in epidemiology? How do we establish causality for relationships that cannot be directly and immediately observed? Epidemiological studies show risk relationships, and we're going to talk about this later in the term in terms of odds ratios or risk ratios, but essentially what we're doing is we're comparing the likelihood or the risk of some outcome. That could be a disease or it could be death related to an exposure. And all of these outcomes are going to be related to something we're calling exposures. So if you think about smoking and lung cancer, smoking is the exposure, lung cancer is the outcome. Before I delve into the specifics of how each of these criteria are used, I just wanted to give an overview of how they relate to epidemiological studies and epidemiological research. So the first criteria, strength of association, has to do with how we measure the outcome. Outcomes in epidemiological studies are measured in terms of odds ratios or risk ratios, and those are simply associations. They're ways of measuring the association between the exposure and the outcome, and strength of association is the concept of how do we evaluate that result? Is it a strong association? Consistency is the concept that different epidemiological studies in different populations will show the same results. Specificity is a concept that really is more appropriate for the laboratory setting. So you should just be aware that it's the concept that a single exposure is only related to a single outcome. So this really only makes sense for a virus, bacteria, or prion. And it becomes important to be aware of it because so many epidemiological results are not specific. So for example, socioeconomic status leads to a number of different outcomes, and a number of different disease outcomes are caused by or contributed to by socioeconomic status. Temporality is a criteria that has to do with our study designs. So when the exposure was measured in relation to the outcome. In particular, we talk about a time relationship, and whether or not the exposure was measured before the outcome has to do with whether or not it was a longitudinal study design or a study design that was carried out over time. One of the criteria that we sometimes use in epidemiology, although not always, has to do with biological gradient. If the exposure has different levels of dose, often epidemiologists will categorize the different levels of dose and evaluate whether that relates to risk outcomes at different levels of exposure. The criteria of experimental evidence is typically used as the gold standard in epidemiology in terms of demonstrating causality. So we talk about a randomized clinical trial as our epidemiological study design of choice because it provides evidence for an experimental design. Now, the last three concepts are concepts that draw on literature from elsewhere that are not necessarily actually directly tested in epidemiology, but they're necessary and important for the arguments and understanding the information that we've gleaned from an epidemiological research study. So once again, the causal criteria, strength of association, consistency, specificity, temporality, time component, biological gradient, dose response, Plausibility, in particular biological plausibility, is something that we use a lot. Coherence, experimental evidence, and analogy. So I'm going to pull each one of these apart and explain more in depth what they mean. I've mentioned how they're measured in epidemiology, but what do they really mean? Strength of association. Well, really the question we want to answer is the association strong or weak? A weak association is what we call near unity. In other words, there's no difference in the risk between the exposure group and the unexposed group. And we measure this using something called an odds ratio or risk ratio. And those are measures of risk comparing the exposed and the unexposed groups. If that number is 1, it means the risk is the same in both groups. They're what we call unity, identical, 1. So the exposed group would have the same risk as the unexposed. There would be no association. So that would be the extreme case. Not only is it's not even a weak association, there's no association. Now, a strong association would be one where there is a protective relationship, so they have half the risk, or an increased risk, which is greater than double. It may not be statistically significant, but these associations would be identified as strong. And a strong association, such as smoking, where they have risk ratios of 15 or even 20 times the risk of lung cancer that are related to smoking, is less likely to be confounded by some other factor. So that concept of confounding, I know I've thrown in a new word, is a concept of confusion. Is there some other variable that might bias our results or confuse our results, like socioeconomic status or other behaviors? Well, when you have risk ratios that show a very strong association, that's less likely to happen. The next argument on the list is the concept of consistency when you're talking about evaluating a causal relationship. Are the results across multiple different epidemiological studies and multiple different populations and over time all consistent? If there is evidence for consistency, that supports a causal relationship because it means that many studies are showing the same pattern of results, and that pattern is shown across multiple populations using different types of study designs, longitudinal or study designs that are carried out over time, cross-sectional study designs that are carried out at the same moment, and also using different measurement tools. Even if the association is weak and there's no strong association, a consistent pattern in the results lends evidence for a causal relationship. So the next criteria on our list is specificity, and I mentioned already, but this criteria really makes sense when you think of a virus, bacteria, or other known biological cause for a very specific disease outcome. So an exposure like HIV, the virus, causes only one disease, the disease condition HIV/AIDS. And the reverse is also true and also important to put into the argument that outcome, HIV/AIDS, is caused only by that exposure. Now, I'm going to give you an example where this really falls apart. So specificity doesn't work when it comes to a number of chronic diseases. Imagine you have an exposure like smoking. Now, smoking causes lung cancer, but it also causes a number of other outcomes, cardiovascular disease, for example. And that outcome, lung cancer, is caused by smoking based on the evidence, but it can also be caused by a number of other things. So it's not specific, both in terms of the exposure, smoking causing only lung cancer, or in terms of lung cancer only being caused by smoking, because there are other causes, including environmental causes for lung cancer, but also genetic. The next criteria is something really important when it comes to making a decision about study designs. A longitudinal study design, which is a type of study that's carried out over time, asks the question, is there an association between the exposure when it's measured before the outcome? Studies that measure both exposure and outcome at the same time, these are called cross-sectional studies, are prone to bias because after a new diagnosis, people will change their behavior, but also people will remember differently. So even if you're asking them to think back in time, they may remember differently depending on whether or not they have been diagnosed with a disease. Next, we're talking about biological gradient. I mentioned this already, but in some instances, an increasing dose or level of exposure will increase the risk, and this can be an important way to look at the relationship. Evidence of a dose response or a biological gradient is used to clarify or support a relationship between a particular exposure, for example, smoking, and the risk of developing the outcome, for example, lung cancer. So if there really is a dose response in this relationship, it would mean that people who smoke at higher levels will have increasing levels of risk. This concept of biological gradient is the rationale for why epidemiologists sometimes categorize levels of exposure to see whether increasing levels of exposure are related to increasing levels of risk. So you're going to see that in the literature. That's the rationale behind it. And just as some examples of that concept of biological gradient, imagine you have levels of cigarette smoking associated with risk of lung cancer. That would mean that those who smoked more cigarettes have a higher risk of lung cancer. Now, this is true. This has been borne out in the literature. Another point is that you could have a higher intake of a nutrient, and instead of increasing the risk, we'll talk about reducing the risk, but it's still the concept of biological gradient. So you could have a nutrient that reduces the risk of an outcome at every level, and that's a different way of supporting the concept of biological gradient. So if you have, for example, greater beta-carotene intake and reduced cancer risk at every level. Just be aware that biological gradient only works if you expect a linear relationship between increasing level of exposure and the biological relationship to risk. Now, sometimes there is a U-shaped or even an inverse U-shaped curve, meaning that maybe the risk will be at the two ends of the dose relationship or in the middle of the dose relationship. So you may have a different biological response that would relate to either a linear relationship or potentially even a U-shaped or an inverse U-shaped risk. An important aspect of the causal criteria is plausibility, and in particular, we often look at biological plausibility, but this can also be whether or not the relationships are plausible, looking at the social, cultural, or psychological mechanisms. I'm going to focus my answer first on biological plausibility because that's an easier, clearer way to understand this concept. Plausibility is about whether the epidemiological relationships shown by the associations make biological sense. Is there a known biological mechanism that relates to the finding? Now, sometimes there isn't, and sometimes because of an association, biologists will start exploring a mechanism and find something that they didn't know. If the relationship is not driven by biology, are there cultural or social relationships that make sense? So it's not necessarily only a biological argument that's used, but typically in a lot of epidemiological studies where the mechanism that is assumed is a biological one, then it makes sense for the focus in the argumentation to be on biological plausibility. I'm going to give an example about plausibility using information that I'm very familiar with on the relationship between in utero or during pregnancy, undernutrition, or other we call insults. And I'm just going to use the example of undernutrition, although it's broader than just undernutrition. It could be infection, other things that cause stress during pregnancy to the fetus. So in utero exposures are related to chronic disease outcomes. So examples were based on the hunger winter in the Netherlands, where famine during pregnancy, especially during the first trimester, was related to later diabetes risk in those infants. Now, the biological relationships between the in utero insults or undernutrition and mechanisms related to diabetes have been confirmed in mice. Now, this occurred only after those first associations were found and people started exploring. The mechanisms have to do with sparing the brain and changes to the organs. So some of the organs related to metabolism are smaller in size, potentially explaining this relationship to diabetes. Other biochemical pathways may also contribute and have been explored, but in addition to the biological mechanisms, there are also social, cultural, and psychological relationships that are also plausible and also known to be related to the impact of famine on individual behaviors, but also on cultural values. So we have a lot of plausibility in the relationship that hunger or famine in a pregnant mother will potentially contribute to chronic disease risk outcomes in her infants. The next argument that I'm going to talk about are actually two different criteria, coherence and analogy, and I'm putting them together because these are very slippery arguments. Coherence really has to do with whether the relationship makes sense with what we know. Typically, we'll build that argument around biological plausibility, and plausibility as a concept may cover this concept of coherence. But sometimes there is something broader about, does this make sense in a general way with what is known? Is it coherent with the information that we've seen, the evidence we have at hand? It is subjective because what evidence counts, and how do we evaluate this concept of coherence? And those are open questions, which is why these are not necessarily criteria that are always used. Now, analogy actually is used quite often. You might see this in a discussion where an association is found, maybe it's a strange association, and the authors of the article are trying to make sense of it, and they may draw an analogy to another relationship that looked similar. So, unfortunately, this point does work against specificity. Again, this is not a checklist. These are different arguments that are pulled together and weighed against each other to build a case for or against a causal relationship. However, drawing on analogies can be really subjective. So if this relationship, if for example smoking causes another type of cancer, somebody might draw an analogy to the fact that smoking contributes to lung cancer, so it makes sense that it would also potentially contribute to another type of cancer. It works against the argument for a specific relationship between smoking and one outcome, but it does provide information that may help us understand the results. The last and best piece of evidence that we use in epidemiology to establish a causal relationship is a criteria called experimental evidence. And there is a particular study design that helps to establish experimental evidence for the causal criteria, and that is a study design called a randomized clinical trial. So we can also talk about randomized controlled trials or interventions, but I'm going to give the example of a randomized clinical trial because I think it's something that most people are familiar with. That's the idea that study subjects would be randomized to receiving either a new drug or to the comparison group where they're given either a placebo or standard practice if there's a standard practice drug. All the study subjects would ideally be blinded, so they wouldn't know which group they were in. Hopefully that would limit what we call the placebo effect, that they would report better outcomes just because they know they're getting the drug. And the researchers, ideally, in a randomized clinical trial, will be also blinded. We call that a double-blinded study. And that means they're giving the drug over to the study subjects and they don't know whether it is the new drug or the standard drug. The drugs are prepared and distributed by subject matter. So the researcher that's interacting with the study subject doesn't know who is getting which, so they can't inadvertently influence the results. So this is ideal because it removes some aspects of subjectivity. It randomizes the study subjects so there is no confusion related to underlying variables. It takes away some of the subjective components that may influence people's answers. And it also is carried out over time so that we can also, in addition to experimental evidence, It establishes a temporal or time relationship. So thinking back to that other causal criteria, temporality, experimental evidence both shows evidence using an experimental study design, and it also, because it's a longitudinal study, demonstrates temporality. So why do these criteria matter? Now, these criteria are actually arguments. It's not a checklist, but these arguments are going to appear in most of the articles you see. So not every argument will be included in every article, but many of these will be included in every article. So how might the criteria actually appear in an article and in the argumentation of the authors? First of all, strength of association is shown when people report the results in terms of a risk ratio or an odds ratio. And typically they will also interpret the results and make a statement if the results are clearly strong or weak. So they may say there's no association. That is a comment on the strength of association, simply to say there is no association. Or is it a weak association or is it a strong association? Is the result found, is it consistent with the literature? And this will be in almost every study that you see. So particularly in the discussion, they will make some mention of comparing their results with the literature and some comprehensive assessment of whether there's a consistency in their findings. Now, specificity of the relationship really doesn't usually show up in an article. It would be rarely used, but might be included for a new type of disease. So, for example, when they're providing evidence that prions is the cause of mad cow disease, that might appear either in the introduction or discussion. A temporal relationship in the results relates to the study design. So it relates to whether the study was a longitudinal study. So this would usually be in either as a strength or a weakness of the study design. So they might say it's a weakness of the study that the analysis was based on a cross-sectional association. That is an argument about temporality, even if they don't mention the word temporality, because the assessment that that is a weakness has to do with the fact that we look for a time relationship. We prefer longitudinal studies. Longitudinal studies help to support a causal relationship. Or they might say this was a longitudinal study, which is a strength of the results. Biological gradient is also something that may not be explicitly there, but it will be there implicitly because it's the logic behind any reporting of result by levels of exposure. So if you're looking at an exposure like socioeconomic status and you see different levels of risk related to low, middle, and high socioeconomic status, we're talking about a dose response or a gradient. So that may not be a biological gradient, but it is the concept of a dose response. Typically, these results will be focused on a biological relationship, like different levels of exposure related to risk. So when you see different levels of exposure and a risk relationship, the concept of biological gradient is the logic behind those results. Now, biological plausibility will almost always be included to justify the aim or to explain the results when there is a biological mechanism involved. If there's not a biological mechanism, plausibility would be supported based on social and cultural mechanisms. So I have emphasized biological plausibility because that's the easiest to imagine, but if they're doing a study looking at socioeconomic status, I wouldn't expect to see the plausibility argument to be based on the biological mechanisms, but more on the social relationships. Finally, experimental evidence will be discussed if the study was a randomized type of trial, whether that was a randomized clinical trial or a randomized control trial or an intervention with randomized intervention and control groups. If there is an experimental type of study design, this would almost always be included as a strength of the study. And sometimes if it wasn't an experimental type of study design, there would be an acknowledgement of that as a weakness, although it's usually only mentioned if that actually occurs. So this is sort of the overview of how the different criteria that we're talking about can appear in the argumentation and how other people use it and how eventually you will use it in your own writing, whether you write an epidemiological study or you simply pull together a synthesis of the literature. These are the things that you're going to be looking for when you synthesize the literature in epidemiology.